Prepare the materials and acquire the patient information needed. Before choosing an appropriate puncture site, make sure that the patient is not allergic to latex and antiseptics. Cleanse the site in concentric manner using wet cotton and allow the antiseptic to air dry. While waiting for the site to air dry, prepare the dry cotton and assemble the two-way needle and adapter. Once dry, reapply the tourniquet properly. Then, insert the needle with the bevel facing upwards and make sure that the needle is properly anchored. Insert the tube following the correct order of draw and fill the tube with indicated amount of blood. Invert the tube to ensure mixture of blood with anticoagulant. Once done, remove the tourniquet before removing the needle and apply dry cotton on the puncture site. Instruct the patient to apply pressure until the bleeding stops. Label the tube with complete patient information. Once done, send the patient and clean the working area. Dispose the materials in their designated waste containers. Next is to centrifuge the sample to separate the serum or plasma and RBCs. Then, make sure that the test tubes are clean and with proper labels. Also prepare a clean pipette with proper label to avoid confusion. Then, carefully separate the serum from packed RBC using the labeled pipette and put it in the labeled test tube. Same goes for the EDTA tube. Separate the plasma using the plasma-labeled pipette and put it on the plasma-labeled test tube. It is important to note that the pipettes should not be used interchangeably. Bring all the materials and specimen to room temperature and countercheck the label and test request form. Remove the test cassette from the sealed foil pouch and label the test cassette with specimen identity on the ID area of the cassette. Then, place the test cassette on a flat horizontal surface. Using a transfer pipette in a vertical position over the sample well, dispense 2 to 3 drops of sample into the sample well. You must read the result at 5 minutes after adding the sample. For the interpretation, positive means 2 distinct pink colored bands appear on test region and control region. Negative is only 1 pink colored bands appear on control region, and invalid is a total absence of pink colored bands on both regions. Record the results and, to avoid confusion, discard the test device after you read or interpret the results. Bring the specimen and test components to room temp if refrigerated or frozen. When ready to test, open the pouch at the notch and remove the device. Place the test device on a clean flat surface and be sure to label the device with specimen's ID number. Fill the plastic dropper with the specimen. Holding the dropper vertically, dispense 2 to 3 drops of specimen into the sample well, making sure that there are no air bubbles. Then, set up the timer and results can be read in 15 minutes. Positive results can be visible in as short as 1 minute. Don't read the results after 5 minutes. To avoid confusion, discard the test device after interpreting or reading the result. If only C band is developed, then it is negative result. If both C and T bands are developed, then it is positive. Bring the specimen and test components to room temperature if refrigerated or frozen. When ready to test, open the pouch at the notch and remove the test device. Place the test device on a clean flat surface. Be sure to label the device with specimen's ID number. Fill the plastic dropper with the specimen. Holding the dropper vertically, dispense 2 to 3 drops of specimen into the sample well, making sure that there are no air bubbles. Then, set up the timer, and then the results can be read in 15 minutes. Positive results can be visible in as short as 1 minute. Again, don't read the results after 15 minutes. To avoid confusion, discard the test device after interpreting the result. For the interpretation, if only C band is developed, then it is negative. If C band and T band are developed, then the result is positive. Lastly, dispose the materials in their proper waste containers. Make sure that the 6-cell black glass slides are clean and dry. Take note that the positive and negative control are prepared separately by our clinical instructor. Drop into separate cell the serum sample and add a drop of latex reagent into the sample. Mix with separate sticks and spread the fluid over the entire area of the particular cell. Tilt the slide back and forth slowly and evenly 8 to 10 times per minute for 2 minutes so that the mixture rotates slowly inside the cell, or place on an automated rotator at 100 RPM. At the end of 2 minutes, read the results under bright light. Compare the results to the positive and negative control. For this test, our sample is negative. For RPR fluctuation test, the positive and negative control are also prepared by our clinical instructor. Drop a specimen and 1 drop of RPR agent. Mix well, spread out the liquid, and gently rock the slide for 6 minutes. After 6 minutes, read and interpret the result. First is to prepare the sample, specifically serum and a VENIPUNCTURE strip. Label the strip with the name of the patient and insert 200 UL of serum in the sample well of the VENIPUNCTURE strip. Make sure that the machine is available for running. Assigning sample IDs to a position manually. Select sample ID, press the arrow keys to input the patient's last name. Select the character and press a number key to place a number in the cursor box. After identification, press the return button to return to the main menu. Load the strips and SPRs in parallel. Run the assay and results are printed automatically. After the assays are completed, remove the strips and SPRs from MINIVIDAS analyzer. And this is the example of the printed results. Prepare a clean test tubes with proper labels. Label the test tubes as follows: TT1 until TT5. For single dilution, place the following value of NSS into the 5 tubes. TT1, place 0.60 ml of NSS. For TT2, place 0.80 ml of NSS. For TT3, place 0.85 ml of NSS. For TT4, place 0.90 ml of NSS. And lastly, for TT5, place 0.92 ml of NSS. Then, place the following volume of serum to their corresponding tubes. For TT1, place 0.40 ml of serum. For TT2, place 0.20 ml of serum. For TT3, place 0.15 ml of serum. For TT4, place 0.10 ml of serum. And lastly, for TT5, place 0.08 ml of serum. Mix all properly. Mix them using the figure of 8 technique to avoid bubbles. For serial dilution, prepare clean test tubes in a rack and label the tubes as follows: TT1 until TT5. For test tube 1, place 800 ml of NSS and add 200 UL of serum to TT1 and mix properly. Always make sure to use separate pipette tips for NSS and serum. For the second tube, place 0.80 ml of NSS and add 200 UL of serum from test tube 1 to test tube 2 and mix properly. For test tube 3, place 800 UL of NSS and add 200 UL of serum from test tube 2 to test tube 3 and mix properly. Same goes for the fourth tube. Place 800 UL of NSS and add 200 UL of serum from test tube 3 and place it to test tube 4 and mix properly. For the last tube, place 800 UL of NSS and add 200 UL of serum from test tube 4, place it to test tube 5 and mix it properly. After finishing all the procedures, you must disinfect the working area.